Hello and welcome to another video. In this video we're going to be talking about normocytic anemias. Those are anemias with the MCV between 80 and 100. So the red blood cells are normal in size, yet these patients are still anemic. And I split them up into metabolic defects and structural defects of the red blood cell because I think that makes it easier to understand. And then I also labeled which ones are intravascular and which ones are extravascular. So intravascular meaning that the destruction of the red blood cell happens within the vessel, and extravascular meaning that the destruction of the red blood cell happens outside of the vessel. I want to start off this video talking first about the intravascular normocytic hemolytic anemias, which would be G6PD deficiency and Paroxysmal Nocturnal Hemoglobinuria. But before we get into that, these are just two overarching points that I think are important to remember in this series of videos. So these normocytic anemias we're going to be talking about are hemolytic anemias. There are also non-hemolytic anemias. But in hemolytic anemias, we have a elevated reticulocyte count. So in all of these, you would see an elevated reticulocyte count, which is your premature red blood cells. And that's because the body's trying to make more red blood cells. But again, there are other types of normocytic anemias that are not hemolytic, but we're not going to be discussing those. Another important point is the osmotic fragility test. And a positive test here indicates that red blood cells lyse more easily under osmotic stress, and that suggests a membrane defect, such as G6PD deficiency or pyruvate kinase deficiency. So G6PD deficiency, this is the name for it. It is most commonly affecting Africans, Mediterraneans, and people of Asian descent. And these are real quick the symptoms that you would expect to see: jaundice, splenomegaly, abdominal or back pain, dark urine, and it's an X-linked recessive inheritance. But it is asymptomatic until triggered. So that leads to the question, what are the triggers? The triggers are fava beans, which are common in Mediterranean diets, infections, drugs that can cause oxidative stress like antimalarials like these, primaquine and chloroquine, and even sulfa drugs. And then diagnostically on histology, you'll see Bite cells and Heinz bodies, which is why you see the splenomegaly. Here's an image of Heinz bodies. And the spleen recognizes these as incorrectly made. It's going to try and get rid of them, and it's going to take bites out of them. That turns them into Bite cells, which you see here. But these Bite cells are the wrong shape, so they clog the spleen and cause red blood cell destruction. And that's where you get the splenomegaly, because the spleen is being overworked, so it enlarges. You can also see elevated reticulocyte count, like we spoke about before, and elevated unconjugated bilirubin, which is why you get the jaundice. And really, there's no cure. You just want to avoid those triggers. The way this works is with this biochemical pathway right here. So normally, you have G6PD, which helps convert your glucose 6P to your 6-phosphogluconate. And that process requires NADP to gain hydrogen and become NADPH. However, glutathione requires that H to be formed over here. And so if we don't have G6PD, NADPH cannot be formed. Glutathione can't become what it needs to become because it can't grab the hydrogen from the NADPH. And so it blocks this reaction right here from happening, where the oxidative stress persists because it can't be converted to water. So our red blood cells normally live in a very stressful environment full of different oxidative stressors, and the body handles them pretty well using glutathione and G6PD. However, when you have certain stressors that can trigger this crisis, then we can't get rid of our oxidative damage, and that causes the red blood cells to lyse and to be destroyed. So that's how that works. And then with our PNH, I think this one's kind of cool. So your normal red blood cells have anchor proteins called GPI. And you can see them here. I drew it out here, but you can also see it here. And GPI holds two things: CD55 and CD59, also known as DAF and MERL. And what they do is they help inactivate complement so that your immune system doesn't mistakenly attack your red blood cell. However, this is a mutation where PIG-A, which encodes for that anchor protein GPI, is not functioning. You don't have it. So you don't have the ability to hold CD55 and CD59 because you're missing the anchoring protein. So that makes your red blood cell susceptible to complement activation and attacking it and destroying it, like you see here. So classically what happens is you have this clinical triad where you have hemolytic anemia. No surprise, because we're talking about different hemolytic normocytic anemias. You have pancytopenia, which means that all of your blood levels are going to drop, right, with your white cells. And then you have thrombosis, because it's also affecting your platelets, right? The pancytopenia is affecting all your cells. You're seeing a drop in all of them, and so your platelets are being destroyed. You have a high risk of thrombosis. So these patients most of the time die of thrombosis. This occurs at night because of the natural complement activation that occurs while you're sleeping. So while you're sleeping, you're a little higher in carbon dioxide. You undergo a little bit of a normal respiratory acidosis that occurs while you're sleeping, just because you're breathing more shallow, a little slower, and that kind of stuff. And all that's normal. But for these patients, that causes this process to trigger. And so you have very dark urine in the morning because of all the hemolysis with the red blood cells. And the only treatment is stem cell transplant to try and get these back in the body, and meds that'll inhibit your immune system. In the next video, we're going to tackle the extravascular normocytic hemolytic anemias, PKD, hereditary spherocytosis, and sickle cell disease, as well as trait.